A patient with chronic leukemia remains unresponsive to standard treatments. In a different ward, a couple experiences their third consecutive pregnancy loss with no clear physiological cause. Neither pathology originates in the patient's environment or diet. The source exists deeper, inside the nucleus of their own cells, encoded directly into the physical structure of their DNA. To read that genetic code, clinicians rely on a diagnostic tool called a karyotype. A karyotype is a photomicrograph, a physical photograph taken through a microscope of a person's individual chromosomes. Once laboratory technicians capture that image, they cut out the individual chromosomes and arrange them by size into a structured diagram. That organized layout is formally called an idiogram. But before doctors could use these photographs to diagnose disease, the scientific community had to figure out what a normal human genome looked like. This diagram illustrates how the Denver system categorizes the genome. It sorts the 23 pairs into 7 distinct buckets, labeled Group A through Group G, organized in strictly descending order of size. The longest chromosomes go into Group A, while the shortest drop into Group G. Within those groups, they are further classified by the physical position of their centromere, the pinched region connecting the chromosome arms. Depending on where that pinch occurs, the structure is categorized as metacentric, submetacentric, acrocentric, or telecentric. To apply this classification system to a living patient, laboratory technicians have to physically extract those chromosomes. The process starts with a standard blood draw. To keep the cells viable, the blood is immediately mixed with heparin, an anticoagulant that stops the sample from clotting. The white blood cells are then transferred into a culturing environment. Technicians feed the cells with F10 media and human serum, providing necessary nutrients. Then, they introduce a chemical called phytohemagglutinin. This acts as a biological accelerator, forcefully triggering the cells to begin dividing rapidly. The active culture is sealed and placed in an incubator, where it is held at exactly 37 degrees Celsius for 3 continuous days. These initial laboratory steps are strictly preparatory. The incubation period functions as a force multiplier, creating a massive population of identical cells. This ensures researchers have a large enough sample size to successfully isolate and identify a single structural error. The actual extraction happens during the harvesting phase, and it relies on precise chemical timing. The process kicks off with the addition of a chemical called colchicine. Colchicine halts the cell cycle dead in its tracks. It specifically arrests cell division during metaphase. At this exact stage, the chromosomes are at their absolute thickest and most tightly wound, making them highly visible under a microscope. Next, the sample is centrifuged to isolate the cells, which are then dropped into a hypertonic solution. This lower concentration fluid forces water into the cells, causing them to swell like balloons. The swelling pushes the tangled chromatids apart, giving researchers a clear view of each individual structure. To lock those separated chromosomes in place, technicians apply a chemical fixative. A mixture of glacial acetic acid and methanol stabilizes the cells, preserving the chromosomes exactly where they are. The final step is staining. Technicians drop the preserved cells onto a slide and apply a specialized dye, most commonly gene-sustain. The gene binds to the DNA, creating a distinct barcode-like pattern of dark and light horizontal stripes across the chromosomes, known as G-banding. Every step in this laboratory sequence is necessary. If technicians bypassed the swelling, freezing, or staining, the patient's DNA would remain an invisible, overlapping mess, entirely useless for any kind of clinical diagnosis. Back at the patient's bedside, the resulting G-banded karyotype is ready for analysis. Doctors read those precise dark and light barcodes to spot missing segments, extra copies, or structural breaks that drive diseases like cancer. This graphic shows exactly what oncologists look for in patients with chronic myeloid leukemia. They scan the karyotype for the Philadelphia chromosome, an anomaly where chromosome 9 and chromosome 22 physically break apart and swap specific segments. Identifying this specific translocation dictates the clinical approach. Patients who test positive for the Philadelphia chromosome actually have a longer survival rate because their cancer responds to targeted therapies that otherwise wouldn't work. This same mapping process provides answers for couples facing repeated pregnancy loss. By karyotyping the parents, doctors can expose hidden chromosomal abnormalities in an otherwise healthy adult's genome that are causing the recurring complications. The diagnostic window extends to patients before they are even born. Using a procedure called amniocentesis, doctors safely extract a small sample of fluid containing shed fetal cells during pregnancy. Technicians culture and karyotype those fetal cells, allowing doctors to screen for severe genetic conditions. They count the exact number of chromosomes to detect trisomies, where a third extra chromosome appears, which cause developmental disorders like Down syndrome and Edwards syndrome. A karyotype is a direct read of human source code. By charting the exact microscopic typos causing a disease, clinicians can make proactive medical decisions that save and